Hello friends, today we will discuss about genetic basis of retinoblastoma and our main objectives will be to give a brief introduction of cancer, to study about the retinoblastoma, to study in detail about the genetics of retinoblastoma, to study about the role of PRB in regulating the cell cycle, to study about the RB1 gene mutation in retinoblastoma. So first and foremost, an introduction about the cancer. Cancer is a disease involving heritable defects in cellular control mechanisms that results in the formation of invasive tumors, capable of releasing cells that can spread the disease to distant sites in the body. Cancer causes about 1/5 of the deaths in the United States each year. Worldwide, between 100 and 350 of each 1 lakh people die of cancer each year. Cancer is due to failures of mechanisms that usually control the growth and proliferation of cells. During normal development and throughout adult life, intricate genetic control systems regulate balance between cell birth and death in response to growth signals, growth inhibiting signals and death signals. In some adult tissues, cell proliferation occurs continuously as a constant tissue renewal strategy. Intestinal epithelial cells, for instance, live for just a few days before they die and are replaced. Certain white blood cells are replaced as rapidly and skin cells commonly survive for only 2 to 4 weeks before being shed. The cells in many adult tissues, however, normally do not proliferate except during healing processes. Such stable cells, for example, hepatocytes, heart muscle cells and neurons can remain functional for longer periods or even entire lifetime of an organism. The loss of cellular regulation that give rise to most or all cases of cancer are due to genetic damage. We will now discuss in brief about the familial cancer. Cancer is such a common disease that it is no surprise that many families have at least a few members who have had cancer. Sometimes certain types of cancer seems to run in some families. Cancers that run in families are known as familial or hereditary cancers. Familial cancers occur in families more often than would be expected by chance. These cancers often occur at an early age and may indicate the presence of a gene mutation that increases the risk of cancer. They may also be a sign of shared environmental or lifestyle factors. Cancer in a close relative like a parent or sibling, brother or sister is more cause for concern than cancer in a more distant relative. Even if the cancer was from a gene mutation, the chance of its passing on to the next generation gets lower with more distant relatives. A classical case of familial cancer is retinoblastoma, which is caused by the loss of function of RB, the first tumor suppressor gene to be identified. Coming to our second objective, that is to study about the retinoblastoma. The first tumor suppressor gene to be studied and eventually cloned is associated with a rare childhood cancer of the retina, that is the light detecting tissue of the eye, called retinoblastoma. Retinoblastoma is a childhood cancer of the eye. Retinoblastoma develops during the period from birth to age 4 years and is the most common eye tumor in children. The gene responsible for this disorder is named RB. The incidence of retinoblastoma follows two distinct patterns. First is that it occurs at high frequency and at young age in members of certain families, and second is that it occurs sporadically at an older age among members of the population at large. There are two forms of retinoblastoma: sporadic retinoblastoma and hereditary retinoblastoma. In sporadic retinoblastoma, in 60% of cases, an eye tumor develops spontaneously in a patient from a family with no history of the disease. In these cases, a unilateral tumor develops in one eye only. In hereditary retinoblastoma, in 40% of cases, the susceptibility to develop the eye tumor is inherited. Patient with this form of retinoblastoma typically develops multiple eye tumors involving both eyes, that is, bilateral tumors, usually at an earlier age than is the case for unilateral tumor formation in patients with sporadic retinoblastoma. The most common and obvious sign of retinoblastoma is an abnormal appearance of the retina as viewed through the pupil, the medical term for which is leukocoria. Other signs and symptoms include deterioration of vision, a red and irritated eye with glaucoma, and faltering growth or delayed development. The fact that retinoblastoma runs in certain families suggested that the cancer can be inherited. Examination of cells from children suffering from retinoblastoma revealed that one member of the 13th pair of homologous chromosomes was missing a small piece from the interior portion of the chromosome. The deletion was present in all of the children's cells, both the cells of the retinal cancer and cells elsewhere in the body, indicating that the chromosomal aberration had been inherited from one of the parents. Retinoblastoma is inherited as a dominant genetic trait because members of high-risk families that develop the disease inherit one normal allele and one abnormal allele. Coming to our third objective, that is to study the genetics of retinoblastoma. The genetic basis of retinoblastoma was explained in 1971 by Alfred Knudsen of the University of Texas. Knudsen proposed the two-hit mutational model to explain the occurrence of hereditary and sporadic forms of retinoblastoma. Retinoblastoma is a cancer caused by two mutational events. One mutation is inherited via the germinal cells and the second occurs in somatic cells. In the non-hereditary form, both mutations occur in somatic cells. This model relates in a general way to all forms of familial cancer. Knudsen's two-hit mutational model, as exemplified by retinoblastoma, is illustrated by this figure. In sporadic retinoblastoma, a child is born with two wild-type copies of the retinoblastoma gene, genotype RB, and mutation of each to a mutant RB allele must then occur in the same eye cell. since the chance of having two independent mutational events in the same cell is very low, sporadic retinoblastoma patients would be expected to develop mostly unilateral tumors. In hereditary retinoblastoma, patients inherit one copy of the mutated retinoblastoma gene through the germ line, that is, they are RB heterozygotes. Only a single additional mutation of the retinoblastoma gene in an eye cell is needed to produce an RB homozygote that would result in tumor formation. As only a single mutation is needed to produce homozygosity of RB, hereditary retinoblastoma is characterized on the average by earlier onset than in sporadic retinoblastoma and by multiple bilateral tumors. According to Knudsen's model, the retinoblastoma mutation is recessive because cancer develops only if both alleles are mutant. However, if one mutation is inherited through the germ line, tumor formation requires only a mutational event in the remaining wild type allele in any one of the cells in that particular tissue. Due to the high likelihood of such an event, the disease appears dominant in pedigrees. Thus, for hereditary retinoblastoma and in hereditary neoplasms in general, inheritance of just one gene mutation predisposes a person to cancer but does not cause it directly. A second mutation is required for loss of heterozygosity. Support for Knudsen's hypothesis came in the 1980s from the analysis of the chromosomes of tumor cells and normal tissues in patients with retinoblastoma. Knudsen proposed that the development of retinoblastoma requires that both copies of the RB gene of a retinal cell may be either eliminated or mutated before the cell can give rise to a retinoblastoma. In other words, the cancer arises as a result of two independent hits in a single cell. In cases of sporadic retinoblastoma, the tumor develops from a retinal cell in which both copies of the RB gene have undergone successive spontaneous mutation. In contrast, the cells of a person who inherits a chromosome with an RB deletion are already halfway along the path to become malignant. A mutation in the remaining RB allele in any of the cells of the retina produces a cell that lacks a normal RB gene and thus cannot produce a functional RB gene product. This explains why individuals who inherit an abnormal RB gene are so highly predisposed to developing the cancer. The two-hit mutation model for cancer explains the difference between familial and sporadic cancers. In familial cancers, one mutation is inherited, thereby predisposing the person to cancer. When the second mutation occurs later in somatic cells, cancer may then develop. In sporadic cancers, both mutations occur in the somatic cells, so such cancers typically occur later in life than do familial cancers because the probability of two mutations is lower than the probability of one mutation. After discussing the genetic basis of retinoblastoma, we will now discuss about the role of PRB in regulating the cell cycle. The human RB tumor suppressor gene has been mapped to chromosome 13. The RB gene was cloned in 1986. It spans 180 kilobase pairs of DNA and encodes 4.7 kilobase pair mRNA that is translated to produce the 928 amino acid nuclear phosphoprotein PRB. The protein encoded by the RB gene PRB helps regulate the passage of cells from the G1 stage of the cell cycle into S phase, during which DNA synthesis occurs. The transition from G1 to S is a time of commitment for the cell. Once a cell enters S phase, it invariably proceeds through the remainder of the cell cycle and into mitosis. The transition from G1 to S is accompanied by the activation of many different genes that encode proteins ranging from DNA polymerases to cyclins and histones. Among the transcription factors involved in activating genes required for S-phase activities are members of the E2F family of transcription factors, which are key targets for PRB. A model depicting the role of PRB in controlling E2F activity is illustrated in this figure. During G1, E2F proteins are normally bound to PRB, which prevents them from activating a number of genes encoding proteins required for S-phase activities, for example, cyclin E and DNA polymerase alpha. Studies suggest that the E2F PRB complex is associated with the DNA but acts as a gene repressor rather than a gene activator. As the end of G1 approaches, the PRB subunit of the PRB E2F complex is phosphorylated by the cyclin-dependent kinases that regulate the G1S transition. Once phosphorylated, PRB releases its bound E2F, allowing the transcription factor to activate gene expression, which marks the cell's irreversible commitment to enter S phase. A cell that loses PRB activity as a result of RB mutation would be expected to lose its ability to inactivate E2F, thereby removing certain restraints over the entry to S phase. The importance of PRB as a negative regulator of the cell cycle is demonstrated by the fact that DNA tumor viruses, including adenoviruses, human papilloma viruses, and SV40, encode a protein that binds to PRB, blocking its ability to bind to E2F. The ability of these viruses to induce cancer in infected cells depends on their ability to block the negative influence that PRB has on progression of a cell through the cell cycle. Coming to our last objective, that is to study about the RB1 gene mutation in retinoblastoma. Mutations affecting both alleles of the RB1 gene represent a prerequisite for the development of retinoblastoma. Heterozygous carriers of oncogenic RB1 gene mutations show variable phenotypic expression. Patients may develop tumors in both eyes or only in one eye because of the variable expressivity. In addition, some carriers show no retinoblastoma at all because the mutations may have an incomplete penetrance. Almost all types of the RB1 gene mutations have been identified, including translocations, deletions, insertions, point mutations, and epigenetic alterations. The majority of germline mutations that have been identified in patients with hereditary retinoblastoma are nonsense or frame shift mutations, mostly located in the exon 1 to 25 of the RB1 gene. These kind of mutations will lead to abortive transcripts that will be degraded, leaving only the transcript of the normal allele. These mutations will have great impact on the phenotype because they completely abolish the activity of one allele. Therefore, the affected patients will most probably develop bilateral retinoblastoma. When a nonsense or frame shift mutation occurs in the last two exons, the transcripts may maintain some residual biological activity which will support that of the normal allele, and this will result in a milder effect in terms of phenotype. Point mutations in intronic or exonic regions cause aberrant splicing and abolish the biological function of PRB. After discussing the familial cancer, retinoblastoma and its genetic basis, we will now discuss in brief about the genetic counseling of retinoblastoma. Whenever unilateral or bilateral retinoblastoma is diagnosed in a child, it is important to consider the possibility of a genetic predisposition and therefore the risk of development of the disease in young children related to the patient. Analysis of the family history and the tumor history of a patient treated for retinoblastoma is essential to evaluate the possibility of a genetic predisposition and the risk of development of the disease in other members of the child's family. It is therefore possible to calculate the probability of relatives of a child with retinoblastoma to present a genetic predisposition to this disease. In retinoblastoma, genetic screening karyotype analysis is the simplest test, providing results within few days of receipt of blood sample. It is the affordable test to most of the retinoblastoma families. Dear friends, this brings us to the end of the lecture on genetic basis of retinoblastoma. Hope you have enjoyed. Have a nice day.